multi-dimensional space is an object or events which we want to study. The objects are described with independent variables. So if we have m objects or observations, each of those objects is described with n independent variables. And all of those data are put in this matrix. So this is our object of our research, is this data matrix. Of course, we can perform different operations in this data space. The most trivial one known from the primary school is the calculation of average value. Of course, one can calculate for each of the variable its average value. And this is a very simple according to this formula. Now, here is one example. We have a multi-dimensional data space. So we have a set of 27 triazoles. These are bioactive molecules. We have activity of those, and each of those molecules is described with 10 independent variables. In QSAR, we often say those are descriptors. So we have 27 objects. Each of the object is described with 10 descriptors. And of course, in this multidimensional space, we can perform further operations. The two most important information about our data, we calculate the variance and the covariance. The variance is defined with this expression. It's somehow called an average, how spread is our variable, and the relation between two independent variables, between two independent variables, is given as a covariance. So we have to calculate those two quantities for our data set. And of course, a normalized covariance is often called correlation coefficients. It's normalized to 1. Let me these covariances and correlation coefficient matrix can be put in the vector if our set has n independent variables. We have a matrix n times n, and this is a covariance matrix, and this is the basic quantity in statistical and chemometrical research. And similar the correlation coefficients matrix. Here is just a very simple example. Our data space with 27 molecules and 10 descriptors, we can calculate the correlation coefficients or the covariance matrix between different variables. For example, first and second, we get very low correlation coefficients, and we can see that there is no correlation here. And for example, for the first and the third, the correlation coefficients is a little bit higher. We can see the graphical presentation here. And this is an ideal case when the correlation is close to 1. So we can see that all points lie on such a straight line. And similarly, when the correlation coefficient is -1, they are on the other straight line. Let me go now to the next topic of my presentation. This is how can we compare how can we evaluate our objects in our data space. So we can calculate distances between all pairs in our data space. And I would like to present these three distances: Euclidean distance, Manhattan, and Mahalanobis distance. Euclidean distance, here is the expression. How to calculate it is just a generalization of expression of the Euclidean distance in three-dimensional space. So if we have more than 3 dimensions, we just make the same formula for multi-dimensional space. There is, of course, how many distances is. If I have m objects, the count of distances is rising with a square of the number of objects, which could be, which is very fast growing. Just a very short example, the population of Slovenia is 2 million. We can calculate the distances between all Slovenian people, but the population of India is 1 billion, and it is too much. We cannot calculate the distances between all Indian people. This is the second distance is the Manhattan distance. The definition is here, is just the sum over absolute value of differences of coordinates. It is very often used in computational algorithms because it's easier to be calculated in comparison to Euclidean distance. Example is here. We have 2 points. The Manhattan distance is just sum over differences. And a property of those that Manhattan distance depends only on starting and final point and does not depend on the path. This is different from Euclidean distance because Euclidean distance depends also on the path. Let me go now to the more complicated case. This is the Mahalanobis distance. Here is what is actually Mahalanobis distance. Here is an intuitive picture. Let me show this example. I have in 2-dimensional space data points like this. And then I add an extra point to this data set and ask, does this point belong to this set or not? Or it is an outlier? And the Mahalanobis distance answered this. For example, in this case, this distance depends on the direction. If the point is in this direction, somehow perpendicular to the data, the distance will be higher, will be larger. If the point is here, the same distance, let me see, the Mahalanobis distance would be shorter. I will show this in the example. But it is maybe interesting to present a historical view on Mahalanobis distance. How does it come? In 1856, mathematician Riemann introduced non-Euclidean geometry, and this non-Euclidean geometry had a big impact on engineering, on machine engineering, and in science, of course. On the basis of this non-Euclidean geometry, 1915, Albert Einstein postulated general relativity theory. It was postulated that the space is not Euclidean, but has a curvature, and the light does not travel along straight lines, but along curves. This is, of course, true from the universe now. And this is due to the gravitation of terrestrial bodies. But about 20 years later, Indian statistician Prashanta Chandra Mahalanobis applied similar principles of non-Euclidean geometry in statistics. He postulated that distances between objects are not Euclidean, but depend on the space metrics, and the variance matrix, the covariance matrix, determines these space metrics. And this was originally published in 1936 in Indian Scientific Journal. So how is the formal mathematical introduction? Is simply if we have a 2 points in a Mahalanobis space, one calculates the distance like a mixed product. Here is vector, here is the inverse of the covariance matrix, and here is again the vector. And this is actually taken from general gravitation theory of Einstein, these expressions. How is this? It's visualized here on the very simple example. For example, we have our original data set, those blue dots. These are our data, and we calculate the covariance matrix and make an inverse of our covariance matrix. So we have our space matrix, and we can calculate now the distances of those red points. So here on these arrows are Mahalanobis distances. We can see that Mahalanobis distances is quite different from, depend on the direction. The second very important method which is built on the covariance matrix is the so-called principal component analysis. I think you already heard about this. This method has been introduced by a British statistician in 1901, Carl Pearson. And how is the idea of this? I will go again to the very simple two-dimensional example. For example, our data points are organized like this along the straight line. So we have our data are described with two coordinates. So our space is two-dimensional, but we can see that the data are very close to be online. So what we are doing, we can rotate our coordinate system in this direction, and so put our axis in the direction of the highest variance. It means in this direction, and of course, in this way, with this rotation, if we rewrite our coordinates, we can see that they are almost all in the one line. We can neglect the second one, and so what we did actually, it was a reduction from two-dimensional space to one-dimensional space. And the generalized for many for multidimensional spaces is this method is PCA. Formally it is introduced with a mathematical transformation. So we introduce new coordinates, which are linear transformation of old coordinates. But what are those elements of those matrix L? These are actually eigenvectors and eigenvectors of the covariance matrix. So we need again a covariance matrix, and we must know some matrix algebra to calculate the eigenvalues and eigenvectors and put those eigenvectors into the matrix L. Then we can perform our linear transformation. How these eigenvectors are organized and what is the meaning on eigenvalues. From matrix algebra, we know that each eigenvector has one eigenvalue, and those eigenvalues count the part of the total variance. So if I go to the our new variable, if I have, for example, 10 new variables, they are organized according to the variance. The first new variable carries the largest variance, and so we can the last new axis carries the very neglected part of the variance. So I can again neglect some of lowest variables, and what I did was the reduction of a high dimensional space to very few dimensional space. And this method is very often used, is a standard method in engineering and science, because we all the time have a large number of variables. Again, this is our simple example. If I perform PCA on this system I shown before, is just a rotation. So our new axes are the first axis is along the largest variance. The third method is self-organizing maps. This is the belong to the family or artificial neural networks. I will present here the structure called the self-organizing map, alias Kohane network or counter-propagation network. This is the same mathematical structure. This mathematical structure has been proposed by Finnish statistician Kohonen, Teuve Kohonen, in the 30s. And how does it work? A Kohonen or self-organizing map is mathematically simply a cube of numbers. It's a cube of numbers, and if I present our organization here, the columns here represent our objects. So the number of those layers here is equal the number of our independent variables or descriptors. So to each of those belongs one independent variable. And each column here represents an object and our object. So we put those objects in this square, and the objects are organized in a self-consistent way with an algorithm which is known as learning or training of neural network. But I won't go into details of this algorithm, and there is a lot of literature on this topic. For example, you can see the description in this paper. But what is important at the end, when our network is trained, our objects are organized in this two-dimensional or in this structure in a way that similar objects are close to each other. Whatever that means, if I look at the top, I see my objects, and they are organized that in a way that similar objects are close to each other. So this is, of course, interesting mathematical structure. We can analyze then clusters in our objects. We can analyze the roles of individual descriptors. We can make a graphical presentation of our distributions. Like here is the color distribution of some property. And so it's a quite a very useful tool for analyzing of multi-dimensional data. Now we have tools, some chemometrical tools presented for classification, clustering, and comparison of data. We have also, let me see, multiple linear regression. I didn't talk about this because I suppose that you are familiar with multiple linear regression. So we can, I will present some views about the strategy of QSAR model development. QSAR is an acronym. Acronym means quantitative structure activity relationship, and this is very special kind of modeling. The models here are built on existing data. How the strategy looks like. This is scheme of the QSAR strategy. So, as I told, the model is built on existing data. So the first one needs a database, a data set of data for a set of molecules. These are often used for molecules. So I need information about molecules, information about property or toxicity or biological activity, whatever I want to study. And of course, sometimes it's useful when I have details about experiment. And what is now our task? First, we have to extract the molecular structure for the database. I need the molecular structures, for example, experimental structure, optimized, or so on, and or some physicochemical properties which will be used as descriptors. This is on the one side. On the other side, I need a uniformly presented toxicity or property. So if I study, let me see, biological activity, I need the same kind of biological activity for entire set of data. For example, LD50 or IC50 or whatever I have, I need this for entire set of data. And this property can be expressed as a number, as a dose, or classifiers. It is classified to, for example, affiliation to particular toxicity class, mode of action, and so on. And then what is my task? I have to calculate from those structures, molecular structures descriptors. These are some parameters. I have algorithms for this. And the QSAR model is a mathematical relationship between those descriptors and property. Of course, there is a lot of ways how to validate and test these models, if the models is good or not. I won't go into details, but if you would like, if you want to develop your own models, it is very useful document published by OECD. This is the Organization of Economic Development and Cooperation, and this organization adopted some principle for validation of QSAR models which are used for regulatory purposes. And you can find this into this web page, and there are some principles and there are some very useful information and instruction which can be used also for development of QSAR models. This is now example of our chemometrical data treatment of a set of conazoles. Conazoles, this is a group of chemicals which are used in agriculture and medicine, veterinary and human medicine, and they are very bioactive compounds. They have quite a, of course, and when they enter the European market or USA market, they must be regularly adopted by officials. So they need the official classification. We have our set, as I said before, 27 triazoles. There are fungicides, herbicides, and some drugs. What we did, actually, we extracted the structure for those conazoles, and then we calculate 10 different descriptors. Descriptors are given in this table. For example, Balaban index, number of cluster counts, exact mass, topological indices, ovality, and so on, shape indices, and also Wiener index and log P, like a property. And what we did, a PCA study. So this is the table looks like this. This color just indicate the level of official classification of those compounds. So our data table looks like this, and we perform PCA study on those data. This is our first results of PCA study. Is called a scree plot. This plot show us the percentage of the total variance carrying by a new PCA axis. For example, our first axis carry about the 80% of the total variance, the second about the 10%, and so on. So our first three axes carry more than 95% of the total variance. I can neglect other 7 axes, so and describe our conazoles in the space of 2 or 3 dimensions. So it's not very good to see, but we can see here some kind of clustering, and also these labels here indicates a different level of official classification of those compounds. This is the PCA1 to PCA3 is the similar classification, and the PCA4. So we can present more picture, and these our labels indicates official classification of compounds. This is was a very bad. Okay, for example, those are substances which are officially classified as a potential developmental toxicants, or not classified, or are in the waiting list, or so on. These are the official classification rules. So the same data can be analyzed with our PCA, with our self-organizing maps or Kohone networks, and we got this classification scheme. So A and B, these are quite a similar status of those compounds. Those which are red are more dangerous. Those with green are not the compounds of concern. But this is our numerical presentation. Let me now go to the second example. This is the viruses, the nucleotide data of viruses. We studied till now Zika virus, and of course, in the last time, SARS, MERS, and COVID-19 viruses. This is the paper about Zika virus, which is already online in a computer-aided drug design. How our working scheme works. First, we have to collect virus sequences from different databases or whatever I will show you later, and then we present somehow those sequences, calculating the descriptors, and then carry out our PCA, Mahalanobis Distance, or self-organizing map analysis of those data. For example, from Zika viruses, we collected data from Gene Bank. The set includes 310 samples taken from different continents, Africa, Asia, South America, and each sequence contains the whole genome sequence of the virus. This is about 10,000 bases, nucleotide bases like adenine, thymine, thymine, uracil, guanine, and they were taken from different mammalian species. Here is the distribution about the countries. It's not very good to see, but I think the largest part is this is the Singapore, this is the Brazil, and the Colombia. For each of those, we have, of course, a total genome sequence. It's about 10,000 bases, and those bases we encoded in our descriptors. We have so that we count simply triples of bases. We have, of course, 64 different triples of bases because there are 4 different bases and 64 different triple of bases, and we count how many of such triples are in individual genome. So we mapped our genomes in a 64-dimensional space, and we performed the PCA cohort network and distance analysis of those space. This is the first example. Now we calculated Mahalanobis distances. We can see that the average value of Mahalanobis distances from Africa samples is twice as large as Asia and South America. So they are somehow separated from other set. And this is confirmed here on this PCA picture. We can see the red area here is Africa samples, and this green and blue is Asia and South America, and they are quite separated. Actually, the Zika virus originates from Africa. He was firstly detected in Uganda, and he spread then over the world from Africa. And this is probably more original version, and these are maybe a slightly modified or mutated viruses. There are some outliers here. You see India is a little bit separated from other, or some samples from Brazil, or samples from Singapore or Brazil, but it's an interesting picture. And similarly, we performed the same analysis using self-organizing maps, and also we got a distribution of samples in such a two-dimensional picture. Let me go to our last example. This is the analysis of proteomics data. Our joint study has been published in already three papers, SAR and QSAR, and current computer-aided drug design journals. What is the problem of proteomics data? The aim of those study was to study the potential hazard of nanoparticles. We know that in the recent years, the application of nano-substances is growing in industry and in consumer goods, and therefore the people are exposed to nanoparticles. And it's of course not clear if they may have some adverse effects on the human health or on the environment. What we know about the nanoparticles, that their physical, chemical, and biological properties of nanoparticles strongly depend, or only depend, on their shape, size, and physical state. which make them, of course, suitable for different industrial application, but it is also not known how dangerous are they for human and for environment. Therefore, it's a part of this research is our proteomic studies. What we mean on the proteome, proteome means actually the information about the status, protein status of the cell, and this status may change if cells are treated with nanoparticles. And here is the quite a big experiment. The details are written here. 3 different cell types were expressed to different nanoparticles under different conditions. So, and each measurement has about 2,000 proteins. So we have quite a big data set. We evaluated those proteomes, individual proteins, making different our different chemometrical analyses. For example, we analyzed the distances, or we analyzed the PCA and self-organizing maps. What was the conclusion? The conclusion was that the most important parameter which determines the cell response was the time of exposure. Always the samples which are under 24 hours exposure time was separated from other samples. And we also tried to include silver nanoparticles in our study, but those data was not complete. So we make just some very simple chemometrical analysis, trying to indicate the most important proteins, but actually we were not very successful here. We can find some proteins, but this is maybe still under investigation. So I think my time is over. I would like to thank actually organizers to give me opportunity for this talk, and I hope you find some new information. Thank you.
Thank you, sir. We have shared your email with the participants. So hopefully if participants have any questions, you can just email sir for your answers. Yes. Thank you, sir, once again.
And you can also share my presentation.
Okay, sir.
Okay. Thank you.
Thank you.
Bye-bye.
Now we are having our next speaker. He's Professor Sukhen Das. He's from Jadavpur University, Kolkata. He will be talking on clay or polymer-based nanosystems for numerous applications. Professor Sukhen Das has a great contribution in teaching as well as in research. He has 30 years of research experience and 25 years of teaching experience. Apart from that, he has received the Best Teacher Award from UGC in the year 2014 to 2015 for his quality teaching. As a visiting teacher, he played his role in different institutions of Europe and America. He has already completed 20 projects from different national and state funding agencies. Till now, 30 scholars have received their PhD degree under his supervision. He has published around 220 articles in various national and international publications. Now, I will request sir to please take over the session. Alok Maiti, sir.
Yes, Sar is yet to join.
Okay, okay. So we will wait for some minutes.
Have gone so unexplored. Scientists
হ্যালো, হ্যাঁ পেয়েছি, আমি ঢুকেছি। আচ্ছা এবার এবার আপনার ঢুকবো হচ্ছে কোথা দিয়ে বলুন তো, একটু জাস্ট বলুন তো আমাকে এটা। আচ্ছা আচ্ছা, আমাকে তো একটু আমি পড়েছিলাম সেদিনকে। আচ্ছা আচ্ছা, আমাকে সেদিন বলেছিল ঠিক।
একটা প্লাস সাইন দেখতে পাবেন, দেখুন
একটা প্লাস সাইন কোন দিকটা বলুন তো?
লেফট সাইডে হবে,
লেফট কর্নার, ডাউন লেফট কর্নার।
আচ্ছা, ডাউন লেফট, হ্যাঁ প্লাস সাইন দেখতে পাচ্ছি।
আপনি ওখানে আপলোড করে করতে পারেন অথবা ডাইরেক্ট স্ক্রিন শেয়ার করতে পারেন। আপনার তো পিডিএফ না পিপিটি?
পিডিএফ, পিপিটি
না, ওখানে কোনো অ্যানিমেশন আছে?
অ্যানিমেশন নেই, অ্যানিমেশন নেই।
তাহলে ওখান থেকে আপনি প্লাস সাইনে ক্লিক করুন, আপলোড।
আচ্ছা, আপলোড দেবো।
হ্যাঁ,
আচ্ছা, হুম,
আপলোড। এবারে ব্রাউজ করুন, দেখুন
ঠিক আছে,
ড্র্যাগ এন্ড ড্রপ আর ব্রাউজ।
হুম, দেখছি দেখছি। আমি তো সেদিন খুলেছিলাম আপনাদের কথা মতো।
হ্যাঁ হ্যাঁ,
দেখছি। জাস্ট a minute. করেছি তো, কিন্তু আসছে না তো।
না, ব্রাউজ করে আপনি উপরে দেখুন এক জায়গায় ক্লোজ আছে, আপলোড আছে, ওই আপলোডটা ক্লিক করুন।
আচ্ছা আচ্ছা, বুঝছি।
আপলোড, তারপরে দেখাচ্ছে দেখুন টু বি আপলোডেড বলে ডট ডট দেখাচ্ছে।
হুম,
ঠিক আছে, একটু ওয়েট করুন, হয়ে যাবে।
ওকে, আমি কি অডিবল?
হ্যাঁ, ইয়েস স্যার, ইউ ক্যান কন্টিনিউ।
ওকে ওকে, থ্যাংক ইউ। Good afternoon, everybody. Good afternoon, everybody. I am Sukhen from the Department of Physics, Jadavpur University. Actually, I just want to describe something which is related to the research for the beginners. In the earlier lecture, it was a very good lecture and very interesting lecture, but my lecture is a little bit different from that. It is just for the beginners. I think you will enjoy that. Actually, when I was working with the, I have started my research work 30 years back. When I was working, I have started my research work. I have no idea. I had no idea what to do and what is research and what in which way I can do that research. So I visited different places and different seminars I have visited, and I have attended some quiz of, related to science. Then I have seen that in a certain quiz, there I have faced a question that what was the best invention of that century, that means the 20th century. Then there was reply from various places, various researchers, different type of reply was there. Somebody said that it is the penicillin is the best invention. Somebody said the DNA is was the best invention, and somebody said the computer is the best invention. Whatever may be, all are very, very interesting and important invention. But from there, I came to just I understand that whatever may be the work we are doing, we should do that work which is quite close related to our society, which will help our society. So application-based work is very, very important. That is why whenever I started my work, and still already as a number of students have completed their PhD under my guidance, I always try to teach them that thing, please do that research which is related to the society. So in this way, I just want to say one thing. At the end of the 2018, I was in the class. There I have seen some patch mark in the leg of one of my students. And after the completion of my class, I called the student and asked, what is this? Then he said that this patch mark is very common to our society, in our locality. Many people have this patch mark. So I asked him, from where you are coming? He said that I am coming from Bantala. Okay. I returned back to my lab and I talked with my student, and then one student just joined. I talked with him, and I said, if you are interested, you can go there and you can search what is happening there, and this may be a topics of your PhD subject. So he went to the Bantala, and he found I just giving you the observation of him. He found that there are basically tannery are there, and the waste of the tannery, waste material of the tannery are heaped in different places, and randomly they are stacking the waste material of the tannery beside the road and other thing. And there was the canal also, and my students said that the color of the water is very black. I think this pollution is the main reason of the skin disease of that student. Then I talked with them, what can we do now? They just went there and they collect the water from the canal and some dust from the tannery waste material, and they analyze them. And after analyzing, they found that the chromium is much present there. Chromium 6 is 20.858% are present there. So I just talked with them whether you are interested to work with this. And this may be, if you can solve this problem, that this may be a PhD work of you. So he gladly accepted that, and he said, yes, sir, we can work in this field. There is no problem. If we can solve the problem, and if we can publish the paper in this field, then there is no problem. Then I give him the green signal. So he started to work. When he started to work, he found when the waste material of the tannery was stored on the ground, then the chromium 6 was leaching inside the soil, and this chromium 6 is very, very dangerous for our health. And dangerous for our health, and chromium-3 is little bit less dangerous for us, but it took lots of time to convert chromium-3. But this chromium-6 is very, very dangerous for us, and this can go inside our body in various ways. This can go inside in our body through ingestion or through absorption of our skin. So we can inhale this chromium inside our body, or through absorption through the skin, it may go to our body. And this chromium-6 in ionic form is very, very dangerous to our body. So what we can do? He planned one thing, and then I didn't sit down and discuss with him. And from there, we found that this is not the problem of our country only. This is the problem of our whole world. Especially in our country, there are three parts where this problem are major problem. That means in UP, you know that UP is very, very rich in leather product. UP, Bengal, and some portion of Tamil Nadu, they are also facing this type of problem, where tons of leather, tons of this waste material are heaped in, stored in the outside on the road and on the ground, and without any purification. They don't purify this. In the developed country, they purified this in the proper way, but in our country, with our business person are not purifying that. That is why it is going inside the soil, inside the water, and the local people are suffering for that. So what can we do now? We planned one thing. That first of all, we have to synthesize and fabricate on sensor, so that we can find out the material. That means what materials are there in the waste material. We have to find out, because one thing is there. We have collected the sample from there, and we have find out the material what is there through chemical test or through atomic exhaustion test, whatever may be. But all are time consuming and price consuming, and this is not so easy. So we have to find out such a fabricated device which can easily identify the presence of the chromium there, and the percentage of the chromium, not only the present, how much material chromium are present there. We have to find out that. So we start to work that. But not only we have to fabricate that, we have to fabricate a device which is biocompatible also, because if that itself harmed some human body in some way, then that will be a secondary problem. We have to solve that problem also again. So we have to fabricate or synthesis a sensor which can sense the presence of chromium very beautifully, and ultimately we have to remove that from the waste material, so that the pollution can be reduced. So when we were searching this, we found something which is very important to us. First of all, we found from the literature survey, we find that boehmite is a very, very important thing. Boehmite is a clay material which is well crystalline material. It is white or pale gray color, and it has a layer-like structure, layer-like porous structure, and the layer between the distance between the two layers is few nanometer, 5 to 7 nanometer. So this boehmite have some property which can be utilized to identify the chromium. So we have done some experiment, and ultimately we decide that we will work with that boehmite. And the boehmite at nano form is very, very important for this work. So we grind the boehmite, and we make the particle size of the boehmite in the order of 20 to 30 nanometer. But this boehmite is not soluble in water. So what can we do to increase the solubility of the boehmite? One thing we have done, that is, we have taken the some gum from the acacia tree. What is this acacia tree? Acacia tree is very common to us for the Bengali people, because actually in Bengali name it is the babla gach. There are some, we find some type of gum from the babla gach, and that is called the acacia gum. That gum can be used to make the boehmite soluble. So what we do, we make a solution of this boehmite, acacia gum, and then we added boehmite in that solution. And after and we heated that and we stirred that for more than 1 hour at 70 degrees centigrade temperature. And after that we dried that and we again grind that and we make a fine powder of this boehmite. And we predict that the boehmite particle which is in the order of 20 to 25 nanometer, and the gum acacia will be absorbed on the surface of this boehmite, because boehmite has layer-like structure and it has a surface charge, and gum acacia also has some surface charge. So this surface charge will attract each other, and that gum acacia will absorb on the, just like in this way. You can see the picture of this composite material. After preparing this powder, we have done some experiment. We have done some experiment just to characterize the property of that material. What is the characterization experiment? First of all, we have done the XID. From the XID, we have find that there only boehmite is present there, and from the peak broadness, we can easily say that the boehmite are in. You can see from this graph that boehmite are in well nanocrystalline form. That means the nano size is in the order of few nanometers, and no other materials are present there. Only boehmite is there, and peak size, peak broadening, indicating the size of this boehmite is in the order of few nanometers. And there is the absorption also. This absorption indicating that the presence of the resin, that the gum acacia, and that is why in the amorphous form. Then we have done the FTR the experiment. From there, we have seen that there are some weak interaction. That is the weak hydrogen bond is present there. From the TEM study, here you can see from the TEM study, we are clear what we have decided. The picture is also saying the same thing. That means the crystal size of the boehmite particle is in the order of 20 to 30 nanometer, and thus dotted form upon the each crystal, that are nothing but the indication of the presence of the resin, that means the gum acacia on the surface of this. And this gum acacia is making this boehmite more soluble in water. Apart from this, we have done some BET test, through which we have find out the pore size, interplanar spacing, and all these things. And most interesting feature of this boehmite is the PL study. PL study means the fluorescence study. One thing is there, boehmite have an absorption at UV range. It is in the order of 370 nanometer. That means if we expose boehmite in 370 nanometer wavelength UV light, then we find a good emission at 430 nanometer. In this picture, you can see this. There is the emission at 430 nanometer. So we find the fluorescence property at 430 nanometer if we expose that in a UV light, which is of the order of 370 nanometer. So this is the basic property of the boehmite which we observe, and this property may help us in solving our problem, that is to find out the presence of the chromium 6, and at the same time to find out the quantity percentage of this chromium also. So up to this, my students have done, and after that they published 2 papers from this work. These 2 papers are very good papers, and it is not that only they are solving the problem. At the same time, they are progressing their PhD work also, because whatever they have found, that is very important for us to solve the problem of our local people, as well as this is very important to publish paper also. They have published a number of paper, or up to the characterization of the boehmite, they have published these 2 papers. I just want to say you this, that this is not only the social work. At the same time, if you do this social work, if you just directed your PhD work or your research work in the proper way, then the research work will be there, as well as you can solve the problem of your local area. Then one thing I have seen, the boehmite have a very good absorbance, fluorescence at 430. If the black line is the absorbance, the lower black line is the absorption, fluorescence of the boehmite without any gum. If we just give boehmite in a water and allow that boehmite to in front of a 370 nanometer UV light, then we'll get this light. But whenever we are adding this gum on it, then the absorption then the fluorescence property increases, and the blue is with the gum. So the presence of gum is helping us in 2 ways. One is it is making this boehmite soluble to water, as well as it is increasing the fluorescence property of this boehmite, and it is biocompatible, and it is completely harmless. After getting this thing, we process again, and what we have found, here is the thing. This is the first thing, is the only boehmite where we allow the 370 nanometer UV light, and you can see that there are fluorescence property are there. But when we are getting this, we just added slowly some chromium-6 in this water, in this solution. And as we are adding the chromium-6, we found that there was no fluorescence property. Fluorescence property is completely gone there. So how this possible? How it is happening? Actually, not possible. How it is happening? So one thing is there. If we have a solution of our powder, that means the boehmite containing resin in a water solution, then if we allow it to if we allow UV light of 370 nanometer to pass through it, we'll find a fluorescence property. But whenever there will be chromium, there will be no fluorescence. So if there is no fluorescence in our sample, then we can conclude that there are chromium in the solution. And if the fluorescence property does not hampered in any way, then we can say that the solution which we are adding in our solution that don't have any fluorescence, don't have any chromium. So from this, we can easily identify, we have calculated this, and from this we have seen that we can easily calculate the 0.00025 ppm order chromium in a solution. So in this way, with the standard chromium available in the market, we have make a calibration curve so that we can identify the unknown sample which we have collected from the Bantala canal or any other water collected from any other process, whether this water have chromium or not. And if there is chromium, then we want to find out the percentage of the chromium there. Now, one thing you can say, one thing strike to me also, that I am talking about the presence of the chromium. Yes, chromium, in presence of chromium, the fluorescence property is reducing. Ultimately, it completely disappears in presence of chromium. It may happen for other material also. It may happen for copper. It may happen for any other heavy metal. It may happen for any transition metal. That is why we have taken a series of cation and anion. And from that cation and anion experiment, where we, in all cases, in our solution, in our boehmite containing solution, we added different type of cation material and anion material. We have seen this fluorescence property is going only in presence of chromium-6, not even in case of chromium-3. In case of chromium-3 also, chromium-3, the property remains the same. There is no change in the property of the fluorescence of this boehmite, but only if we add chromium-6, then only this property is going away. Otherwise, the fluorescence property will remain the same. So from here, we can easily conclude that we can identify the presence of chromium-6 heavy metal in the water, in the solution, and no other metal or any cation or anion can do this work. So if the fluorescence property reduces or completely disappear, then we can say that material which we add in our solution is containing chromium-6. Now, how this reaction is happening? This reaction is happening whenever we are exposing boehmite in 370 nanometer wavelength, then the charged particle transit from lower ground state to excited state, and whenever it is going to the excited state, it is returning back. When there is only boehmite, then what is happening? Whenever we are exposing it in front of the 370 nanometer when the light, UV light, then now it goes the particle goes to the ground state to the excited state, then it return back, and when it return back, then it emit light of 430 nanometer. So we find the fluorescence property. But whenever we are adding some chromium there, what is happening? Chromium have a polarity, so that also block the surface of this boehmite. Also, it can go inside the different layer of this boehmite particle. So it can go inside the boehmite particle. So in presence of 370 nanometer UV light, when it's excited, then charged particle go to the excited state, but that in that excited state, it will transfer its energy to the chromium-6. So the charged particle, excited particle will return back in another way where we don't find this chromium transition. So that is why in presence of chromium we don't get any fluorescence property, but in presence of any other anion or cation, this does not happen. So this is in that case we get this property. Am I to stop here, Didi? Hello. Am I to stop here?
No, sir. You can continue.
Okay.
Time is there.
Okay. So till this experiment was done, we are doing with the market the chromium available in the market. Now we started our work to standardize with the unknown material, the unknown sample which we have collected from the Bantala. Here you can see the black line is indicating the absorption spectra of the boehmite containing acacia gum. But whenever we have added that water which have collected from the Bantala canal with the fluorescence property absolutely gone, there is no fluorescence property, just which we have found in case of the chromium addition when you have done this experiment in our laboratory. So from this experiment we can say easily that this material, that this water which we have collected from the Bantala contain chromium in a large amount. Now the sensor which we have developed with the help of boehmite and gum acacia, how much biocompatible is this? How much eco-friendly is that? Actually, generally we inhale that chromium through our nose or mouth, or it goes to our body through the skin. So we have taken the lung cell line WI38 lung cell line we have taken, and we have experiment. We have done on this on the lung cell line our sample, which is boehmite dope containing resin. And we have seen from the cell line experiment that it is completely safe up to 100 microgram per milliliter. So we can say that our material which we are using to identify the chromium in chromium presence is completely biocompatible, as well as we can study we have done the morphological study. There we have also seen on the first day to 7th day, there is no change in the lung cell line. So from this we can easily say that that our product is completely biocompatible and eco-friendly. Now, at a glance, what we can see. In first case, we have taken only water, and if we expose that in 370 light, 370 nanometer UV light, it will be completely dark. But if we add some powder which we have prepared with the help of boehmite, whose particle size is in the order of 20 nanometer to 30 nanometer, and some gum acacia resin was added there, then if we add that, we will get a fluorescence property like this. If when we will expose that in the UV light of 370 nanometer. But if we add chromium or that liquid which we have collected from the Bantala canal, then again the fluorescence property will go away. So it is a very clear indication from which we can easily find out the presence of the chromium, and as well as we can easily find out the percentage of the chromium if we make a calibration curve. Now, if we calculate the kinetics of the absorbance, how the absorbance is taking place, then we find that the absorbance is taking place within a very short period, within the 3-4 minutes, it will go to the saturate point. After that, it will remain in the saturate point. So there is no problem. It will not be a very time consuming, and up to 80% chromium will uptake within the short time. So this is the actual schematic diagram. This is the chromium have this layer-like structure, and upon that this black spot are nothing but the gum acacia, which is making this soluble in water, as well as due to the presence of this gum acacia, the fluorescence property of the boehmite increases. But whenever chromium is added there, chromium is also attached on the surface of this boehmite material, or it go inside the stack. And whenever it will go inside the stack, there will be a contact between the boehmite particle and chromium particle. So when it will be this type of material will be exposed to 370 nanometer UV light, then this chromium will take part in the transition, and the transition will take place in the another route, and that is why we will not get any transition in any fluorescence property in the solution. Now one thing is there. We have done all these things to identify the chromium and other things. Now one thing is there. This is little bit time consuming, and not only that, to do this we need a PL instrument, that is the fluorescence property instrument, so which is costly, and for that, and this fluorescence instrument is not portable, so we have to go to laboratory to find out the presence of the chromium and the percentage of the chromium. That is why up to doing this, we have tried to make a easily portable and low-cost detector which can easily detect the presence of chromium and the chromium and percentage of the chromium. And in the earlier experiment, one thing I have forgotten to tell you, that whenever this chromium will stack in the boehmite, then if we can shift this properly, then we can easily eliminate the chromium with this boehmite. So that will be a pure water without the chromium. So it is not very difficult to do that. We have done this in our lab also. We have separated this boehmite, and with this boehmite, with the chromium is also separated from the water, and it is in this way it is possible to separate the chromium from the polluted water. Now, to make a solid cell, we have done one thing. We have taken PVDF, that is polyvinyl fluoride. Polyvinyl fluoride is a polymer which is very commonly used polymer. It is not very costly and easily available polymer. We have make a solution of this PVDF solution. PVDF is soluble in DMSO. In DMSO, we have make a solution of this PVDF, and in that I have given our dust. That means the boehmite added with the gum acacia. And from there we have made a thin film, and in that thin film we have made two lead. One is positive and another is negative. And whenever we have connected this through a resistance box, resistance meter, we can find some resistance there. Now, if we put this thin film in the chromium containing water, in this graph you can easily see, initially this is without any chromium contamination, and this is with chromium contamination. So when there will be no chromium contamination water, then the resistance will be very, very high. But whenever we will dip this in the chromium content water, then the resistance will decrease. So in this way, if we can make a calibration curve, then we can easily find out the percentage of the chromium also. And this resistance meter is not very, very costly and very big one. So with the help of this, we can easily identify the chromium percentage in any way, anywhere. We can take this. This is biocompatible and low cost. So we can easily solve this problem very easily with the help of this solid cell, through which we can find out the presence of the chromium and as well as the percentage of the chromium. For this, we don't need any fluorescence diffraction pattern. We can use the resistance property of this chromium, of this cell, and from where we can easily find out the presence of the chromium and the percentage of the chromium also. And this is very low cost, and this is flexible because polymer is flexible, so you can easily, there is no question of any breaking and any other thing, it will not destroy. And you have seen that we can reuse this, because whenever we are pouring, we are inserting this cell inside the water, then the resistance will increase. Whenever it will decrease, or whenever it will remove from the water, the resistance will decrease. So in the increase. So in this way, we can use a solid cell several times without any problem, and it is biocompatible as well as flexible and low cost also. So we can easily use this to find out the presence of the chromium and percentage of the chromium. So now, if we look into the thing, that this part is the left-hand side part is suitable for the solution, and the right-hand side is for the solid cell, to find out the weather. So in this way, two things our students have done. One is they tried to find out the presence of the chromium and eliminate the chromium from the solution in the solution form. And they also develop a solid cell through which they can easily find out the presence of the chromium and the percentage of the chromium. And both are biocompatible, both are low cost, costly, and eco-friendly, and there is no problem there will be no problem to find out this thing. And my student with whom I have started my work, he has gone to the Bantala and demonstrated this, and they have given some hard solid cell to them also to identify whether there is number of what is the percentage of chromium their presence and other thing. So this cell we can use in our drinking water system. We can use this type of material in the boehmite particle to purify the river water and all these things. Even in our blood, we can find out the percentage of chromium. We have seen the person of the Bantala. We have tested their blood also. We find that they are in the blood, their chromium content is much more than the normal condition. So this is very helpful for the industry belt oriented area, especially where the leather industry, iron industry, and all this type of industry are there, where chromium is a major factor. This is our future plan. My student is doing his research work. He has already published 7 papers in this work within the last 3 years. He has published 7 papers. He is on the verge of the completion of his PhD work also. I am just giving him, trying to, this is a very, very easy work, but this is very important also. So my request to the new researcher that you can take this type of problem which will help our society, as well as at the same time you will be able to complete your PhD or initial work of Java, or I can request the faculty member also, so they can give this type of problem to their student for the PhD work also. So thank you very much, and I am again thankful to the organizer for giving me the opportunity to say something about our research work, and I am glad to get a chance here. Thank you very much.
Thank you, sir. Thank you for your nice presentations. It will encourage us.
Sir, we have one question.
Yes.
Chat box. Is acacia gum can be used with other nanoparticles like alpha or beta nano iron oxide?
Actually, I have to see, because acacia gum we have used just whenever we have found, actually, yes, it was a trial and error method, and moreover, we have gone through the different type of journal also. From there, we got the idea of boehmite. And boehmite, their clay material have huge potential. So boehmite but boehmite was not soluble to the water. That is why again we start to study what can be done. Then we have found the idea of the gum acacia. Then we use the gum acacia, and we have seen that it make the boehmite soluble. And the additional achievement of us is that the presence of this gum acacia is increasing the fluorescence property of the boehmite. That is also very important. It was not in our calculation that the fluorescence property will increase, but we are fortunate that the fluorescence property also increases, as well as it make the boehmite soluble in water. I have no idea. It may be, or I think it may be, because gum acacia have a surface charge, and if there is surface charge, then it can make other molecule to soluble in the water. But I have to do some experiment.
There is another question. How You are differentiating between no chromium with the addition of chromium. Number of chromium, I think so, with the addition of chromium.
How?
Sir, can I ask?
Yes, yes.
Yes, you can
My
ask you.
question is that, sir, you are, as per your data, you are showing that no chromium, there is no fluorescence.
Yes.
There is still water, and after that, and with the,
No. When there will be no chromium, there will be fluorescence. But with the addition of the chromium, the fluorescence will go away.
So this go away, and how we can say, suppose in the some areas water does not contain chromium.
Yes.
Then while its addition of this acacia and boehmite is not giving fluorescence, or already there is no chromium, how we can differentiate that?
No, if there is no chromium, then fluorescence will remain there. If you expose that in the wavelength of 370 nanometer, the fluorescence property will remain there. If only the chromium will present, then actually what is happening, as I've shown you earlier in this graph, this is the pathway. This is the pathway. As I have told you, I have tried with all these things. Only chromium can reduce the fluorescence property. In any cation or anion are not successful to reduce the fluorescence property of the chromium, of the boehmite. Only even chromium 3, this green line is for the chromium 3. Chromium 3, if we add chromium 3, then also there will be fluorescence property. Only if we use chromium, then the fluorescence property will reduce. This is the thing, what is happening. Whenever we are exposed that in presence of 370 nanometer, it will go to the ground state from boehmite. It will go from the ground state to excited state, and it will return back at 430 nanometer. But
no, sir, my question is not that, sir. My question is, suppose there is a pure water is there, so there will be no fluorescence.
No, there will be no fluorescence.
And after, when your material has fluorescence, and after adding the chromium, then only fluorescence will be disappearing.
Yes.
Suppose I am testing some places water where there is no chromium.
Yes.
So it will not give any fluorescence.
No.
But then
here, if you look into this, this is the pure water. This first one is pure water. There is no chromium, no gamma acacia, no boehmite. So if you expose that in presence of 370, there will be no fluorescence.
Okay.
But if you add the boehmite and gamma acacia, then there will be a fluorescence like this. So in pure water, there will be no fluorescence. Only if you add the boehmite and gamma acacia composite, then only you will get this fluorescence. And if you add any other thing there, and if fluorescence disappear, then you can confirm, you can say that contains the chromium. If there is no chromium, it will remain the same as the second line.
Okay.
The first line is with the water only. There is nothing, no chromium, no boehmite, no gamma acacia. Second line is only boehmite and gamma acacia is present in the second line, and in third line we have added chromium, and it remains this one.
Another question we have.
Yes.
How is the regeneration of the biosensor done?
Regeneration of biosensor.
Yes, sir.
Yes, it is where you can use this biosensor for several times, because it will not disturb whenever, as long as especially in the solid form, in PVDF matrix, there will be boehmite containing boehmite, and on the surface there will be gamma acacia. So fluorescence property will be there. Whenever they will come in contact with the chromium, then only they will the fluorescence property will directed in another pathway, will flow in another pathway. So the fluorescence property will go. Whenever we will remove that from the chromium containing water, then again it only by boehmite and gamma acacia will be there because it is embedded in the matrix of the PVDF.
Sir, but you have stated that the chromium ions are being absorbed on the surface of this biosensor.
Yes, whenever there was boehmite will be free from the PVDF. Then the chromium will able to go inside the interlayer, interstacking layer, because it is a layer-like structure. It is a layer-like structure. Whenever you will allow the chromium to go inside, but whenever this boehmite will be in the matrix of the PVDF, in PVDF, then it will not be able to go inside the interlayer structure. It will just touch the surface. So in that case, if the fluorescence property will not go completely, it will reduce in presence of chromium. And whenever you will remove that, and if you wash that with the water, the surface absorb chromium will go away. That is why we can say that the solid material is reusable. You can easily reuse several times there. We have tested that in our lab also. But it is not reusable when you use that in the powder form in the liquid. Then you have to eliminate, you have to wash out that, and with that boehmite, the chromium will also go out.
Okay, sir. Thank you.
Sir, so if anybody, please email sir. Yes, so your email ID, we have provided it in the chat box, so they can just put their queries to you in email. So all the participants would put your queries in email.
Okay,
So thank you, sir.
Thank you.
Sir. For the participants, we will be ending the session here, and next session will start from 7 p.m. So please join in time, before 10 minutes before or 15 minutes before.